It's really my pleasure to be here to host the Yale Climate Day this year. My name is David Vasseur. I'm a professor in ecology and evolutionary biology. And Yale Climate Day has been a tradition on campus. I don't actually know how far it goes back, but it goes back a fair number of years. And for those of you who have attended before, I think you realize that often it is a showcase of the expertise we have here in-house at Yale University on climate-related issues stemming from the physical sciences through the biological sciences and the social sciences as well. Today's going to be a little bit different. We've got an amazing slate of outside speakers who are here today, and that's in part because of a joint venture that we've been able to put together through a Planetary Solutions seed grant and the generous support of YIBS to run this event. So what it was 8 or 9 months ago, perhaps a little longer, Carla and Eric asked me if I would host Climate Day. And of course, my knee-jerk reaction was to say, of course not. But it happened that Martha Munoz and I at the time were putting together, or had recently put together a Planetary Solutions grant to bring people here on campus to think about thermal performance and thermal performance as it pertains to risk of species and risk being very openly defined, I think, still at this point. And we realized we had a unique opportunity perhaps to dovetail these two events. And so we've been able to put together what will be a working group for the next 2 days, but to use the expertise we've brought in through the working group to be able to offer a different slate of speakers for Yale Climate Day. So Yale Climate Day this year has more of a theme than we've had in past years. We are going to talk today mainly about this concept of thermal performance or thermal performance curves and thinking about how they may or may not improve forecasts of risk. That's my generic title for this, and obviously there'll be a little bit more latitude given to the speakers today. I want to say a big thanks to my partner in crime, Martha, who is running the working group and has been a partner today in getting the slate of speakers involved. And also to Carling Beeg and Allison Roby, who are members of my group, who have been great with the logistical support for our event and are going to be continuing to partake in the working group and support the event that we've got going on through the rest of the week. So I'm going to start with a short preamble, and I will keep it short because my work is not part of the slate today, but I wanted to introduce perhaps a bit of a common framework and some common language to what we're going to discuss here today. And the focus is, of course, on thermal performance. So thermal performance curves typically describe some aspect of performance on temperature. And when we think about performance, we might think about traits like swimming speed, other kinds of rates, or even things like fitness. Typically, these curves are unimodal and left skewed. And for the most part, we're talking about ectotherm organisms. There are thermal performance curves for endotherms as well, although I think most of that will fall outside of our expertise here today. Often we wonder about why these curves take on this shape. And there are some great hypotheses underlying this. And if you go down all the way to the molecular level, there's a clear comparison here between the rates at which chemical reactions, biochemical reactions accelerate with temperature, and the stability of things like proteins and enzymes in these systems that drop off sharply at some critical temperature. And so you can take the product of those two components and arrive at a curve that looks something like this. And we often use that sort of invocation to describe why we see a shape like this for most thermal performance curves. A little bit of terminology or jargon, perhaps that might be useful. We often discuss some of the aspects of this curvature, and some landmarks on these curves are things like CT-min and CT-max, which represent critical thermal limits where a particular function or performance reaches zero or ceases to happen. We also have things like an opt, sometimes CT-opt, sometimes T-opt. In fact, I'm going to sort of introduce the idea that sometimes we drop the C entirely, and sometimes, or there's a bit of a motion in the literature to perhaps organize this a little bit more, and I'll present this today and we can perhaps discuss, that when we think about fitness or aspects of fitness, sometimes we tend to prefer this concept of Tmax and Tmin to keep it separate from physiological or rate-based traits. And I'll give you an example why that becomes important. If you think about some aspects, negative values are certainly possible. If I convince you that I can plot something like Malthusian fitness, the population growth rate, then certainly negative values are allowed. But of course, if this is a physiological trait or rate like a swimming speed, then negative values make no sense. So we have already sort of a break in terminology and usage that exists here that is important to think about. One of the real challenges we're faced with when we think about thermal performance curves is how they scale across different levels of organizations. So this is an example of a bunch of different curves for things that are sort of the basal side of this rates. So this is the rate of CO2 uptake, speeds, right? Physiological traits or performance traits. And then on the very end here, fitness. And obviously these things are connected within individuals, but drawing out these connections is a very complicated process, right? And in fact, these curves from this particular analysis are all from different species. There are very few examples where we can connect from rates through performance traits through to fitness within the same organisms. And that's one thing that's certainly important as we think about sort of unpacking some of the challenges with this. Now, I mentioned that this was going to be somehow discussing risk, or we were going to focus on this question of risk at some point. And I want to just give some examples about how we often think about risk and perhaps why that is flawed and where some advances are taking place right now. So one of the ways we do this is to think about things like ambient temperature and how ambient temperature changes through time. And we may be able to superimpose a distribution of the temperature that an individual or a population encounters over its lifetime and ask a question that is simply, how well do these two things match up? And in fact, this is an approach that's been taken in a number of papers, one of which is, I've included my own here on the very right-hand side, but some of this idea comes, or some of the ideas around our notions about which species may be most affected by climate risk or climate change-induced risk are examples that come from here, like the Tewksbury paper, where we see that tropical species tend to have much narrower thermal performance curve and therefore are at risk of small increments of change having a much larger impact on their function, on their fitness, compared to, say, temperate species. And we see that pattern was confirmed in a paper by Curtis Deutsch here in the middle. And then I published a paper where we took a different tack. We looked at climate data on a different resolution that included more day-to-day variability in those estimates, and we see a change in that pattern. And so this highlights some of the importance of timescale and some of the finer considerations in how we approach these kinds of comparisons. If we go back to these thermal performance curves, there are some standard metrics that people often use to measure risk. We think about things like the ambient temperature or the mean of the ambient temperature distribution and its relationship to the optimum for a particular species. And this is sometimes called the thermal safety margin, although again, this can be defined in various different ways. We think about things like the warming tolerance or how far the ambient environment is from this critical maximum. And when we do this, we often end up with examples that look something like this. So this is a nice paper that looked at a variety of different marine species in coastal environments or in tidal pools that are landlocked. And you can see there's a huge difference between those two different groups, the pools versus the coastal environments. And you can see then with a current versus a 2100 sort of forecast what the danger is for those species. So negative thermal safety margins mean that the ambient environment is already surpassed T-opt for those species. Right? So this is a nice metric of risk. It's one of the ways we can capture risk using things like thermal performance. It's also worth recognizing that sometimes temperatures exceed CT max. Right? And how often and for how long this happens can have really detrimental consequences for species. And it depends upon how lethal this is to individuals. And this is an area, in fact, that I think a lot of the recent development is really starting to focus in on. How often, for how long are these sort of moments of extreme stress for species, and what are the consequences of that? Of course, we can break this down further to think about what really happens at the individual level. And we know that ambient temperatures are not a great representation of what individuals actually experience or what their body temperatures actually are, because individuals have different biophysical properties, different albedo. They can behaviorally thermoregulate by using variation within their environment. So in fact, we might have a different distribution of the operative temperatures that individuals experience. And that can change then our perspective on what a thermal safety margin or a warming tolerance might be for those species. So important ingredients to consider. And the last slide I'm going to show here with a thermal performance curve on it is the one that catches everything else. What I might call historical or legacy effects, the effects of acclimation, of evolution, of other stressors like pH, oxygen concentration, nutrients, competitors, predators, all of which have the capacity to reshape the thermal performance curve of a focal species. And this is again an area where a lot of recent developments have been made, and we're starting to learn about the interactions that temperature has with these other factors, giving us some capacity to understand how the multiple stressor, what I'm calling the multiple stressor effect, but really sort of the more holistic picture of ecology and evolution really encompasses. So I think I tried to sum this up with a flow chart, which is always a dangerous thing to do because this is overly simple, leaves out many of the most important factors, I'm sure. But you can think of this as sort of a connection, right? And the connection is here sort of a hierarchical and ecological connection. We're missing the evolutionary feedbacks, the plastic feedbacks that might exist in this picture. But really, one of the things we try to do in this space is to think about how environmental temperatures, the spatial and temporal variation in those, connects through to elements of fitness or elements where we can prescribe some metric of risk. And I would say really those come in three different categories. We can think about risks of changes in body condition or not undergoing maturation through a specific life history stage. We can think about risks at the organismal level, so death of individuals, changes in lifetime fecundity, or we can think about risks at the population level, right? Or species level, extinctions. And so connecting this framework across these different levels, I believe is a goal that many of us are trying to achieve. The talks you're going to hear today, I think, give us a different, perspective on what elements different individuals are trying to cover. And perhaps someone's trying to cover them all. That would be fantastic. I myself tend to short circuit this graph. I'm interested as a population ecologist in the effects of environmental temperature, and I leave most of this information out. So it's really exciting for me today to have a group of speakers that are going to fill in some of these missing gaps in my knowledge and understanding of how this all comes together. So that's my quick preamble. Here's the schedule for today. So we're going to have 3 talks this morning, and then we'll transition into a nice long lunch where we'll have lots of chances to talk with the speakers and amongst one another. And we'll come back for more talks this afternoon at 1:30. We have a fantastic poster session and photo exhibit that's going to be set up here around 3:45. There will be food served and a bar open at that point in time. So I hope everyone is available to stay around for the afternoon and participate. So I'm going to transition over now, and I'll invite Ray Huey to come on up and plug in here, and I'll give a quick introduction while we change over. I'm going to keep my introductions today brief because you're not here to listen to me introduce speakers, but I do have to say a few words. And first, to start by saying what a pleasure it is to have Ray Huey here today to start off our event. Ray is a pioneer in the areas of behavioral thermoregulation, ecological and evolutionary physiology, and I'm probably, not doing justice to your work to just try to bin it into those 3 categories. Your work has been so incredibly influential that I think many of us here today can say that we've relied on data, theory, ideas, concepts that have come from many of your papers. You're visiting us today from the University of Washington, where you're a professor emeritus. And I've just heard, actually, through Martha this morning, that you've been recently elected to the National Academy of Sciences. So congratulations. Yes, indeed. So thank you. With that, I'm gonna pass the floor over to you. So thank you.
Thank you. I just learned that yesterday on the plane, so still a little flying high. First of all, I want to thank David and Martha for the honor of the invitation to join you here and for the opportunity to interact with a number of first-rate biologists over the next several days. And as I was putting together this talk and trying to think what I was going to talk about having to do with thermal performance curves, I had the rather disturbing realization that I made my first empirical thermal performance curve a half a century ago. I find that really disturbing. And at the same time, I was working on a mathematical model with Monty Slatkin. So happy golden anniversary, at least for my role in thermal performance curves. So I want to talk a little bit about how I got started in the field. When I was a beginning graduate student in the late 60s and trying to search for a thesis topic, these were the areas that I thought were really interesting and hot. And physiological ecology, I think, was not on the map in ecology or evolution. Certainly in aquatic systems, that's not true, but at least in terrestrial systems with like lizards, it was not being done. And I got interested in thermal biology as between a master's and PhD when I had the opportunity to spend 10 and a half months in the Kalahari Desert with Eric Pianka. And we camped out. The Land Rover had neither a heater nor an air conditioner. So I experienced seasonality vividly. And it was an exciting place for a young biologist to learn biology, to have an immersion experience for so long. And Kalahari was a great place to just wander around looking for lizards. There were some amazing ones. This is a lizard that mimics a beetle. David Attenborough has filmed that. This is not tan, that's just dirt. We got cold showers about once every 2 weeks. It was not all fun and games. There were some hazards of working in the Kalahari. Whoops, go back one. A lot of venomous snakes. And if you look carefully, at this point it was, you'll see 2 eyes belonging to a lioness. So there were leopards, hyenas, and lions running around, which made hunting for lizards spend a lot of time looking over our backs. In any case, the experience was formative. As the seasons went by, I noticed that the behavior of lizards were changing. If you look at this agamid lizard in the summer months, it was active only early and late in the day. It was missing the hottest part of the day in the summer. But in the winter, if they were active at all, they were active only on warm days, sunny days. They're active only in the middle of the day. It was clear that their activity times were driven by the thermal environment. If you look at where the lizards were found, it was obvious too that they were changing microhabitats as the seasons went by. The escape was, in the summer months, was up in the trees, in the shade, off the hot ground with higher wind speeds. In the winter, they move out onto the ground where they can bask effectively and the wind speeds are lower. So I left the Kalahari thinking, yeah, thermal biology is important, so what? And then I had the opportunity when I came back and restarted graduate school, had the opportunity to spend several summers in the Caribbean working on Anolis lizards. And one of the species I worked on is Anolis chrysotellus in lowland Puerto Rico. And I worked in 2 habitats. One, there's an open park where the lizards live on the lower parts of the trees. They can easily bask and get shade if they want there. But I worked also in the adjacent forest just a few meters away where there's very little sunlight getting through. It's filtered. And I looked at the behavior of these lizards and temperatures. And they were remarkably different. The ones in the open forest, open habitat would bask early, so you get a fast rise time in body temperature and a relatively constant temperature during the day. Whereas the ones in the forest were thermoconformers. So these are just a few meters apart. And I started wondering, what does it mean to be active at 24 versus 32? How does that affect the ability of lizards to do whatever they're attempting to do? And I was sort of intrigued by that, especially because of the differences here from what I had seen in the Kalahari, where lizards were thermoregulating carefully. Here, some of the ones are thermoconformers. And that led me to think about developing a thermal performance curve, which David has introduced. And I realized, oh, if I could just measure some trait at different temperatures, fit a curve to that, I could easily estimate some metrics that would be useful in translating body temperatures from the field to some level of performance. The question was what to measure. My first thought was locomotion, because that seemed to be important to the ability of a lizard to capture prey or to escape a predator. Incidentally, in July, I'll be backpacking in the Lamar Valley of Yellowstone, where there are a lot of these. So I'll learn firsthand about escaping. In any case, in the early 70s, there were no microcomputers. There weren't even cell phones, believe it or not. And so how do I measure speed and acceleration? It was a challenge. But my office mate had a frog and a yardstick. And so one Saturday in the basement of the biolabs at Harvard, I jumped a frog at a series of temperatures, fit a curve, and that was half a century ago. Incidentally, one of the botany graduate students walked by and watching me on the floor jumping this frog and said, "Well, Ray, looks like a high school science fair project." I have to agree, but I got a lot of mileage out of that frog. Okay. Then when I got to Berkeley as a postdoc, I had the good fortune of working with a very talented psychologist who helped me design and build effectively a computerized drag strip. So you can't really see it, but there's a series of photocells down the track. So you can chase the lizard down it and measure acceleration and speed. And when I did that, this is what I found for Anolis cristatellus. So now I can take the body temperature data that I'd collected previously. Now I know how temperature affects performance. I can combine those two together. And that's shown basically here. You tell me a temperature, I can predict performance. And so this is what I was getting for the lizards in the two habitats. What was surprising to me at the time was that lowland Puerto Rico, at 6 o'clock in the morning, 24 degrees, that's warm for these lizards. They're already running at 90% of maximum speed. The open habitat lizards are too hot probably in the middle of the day. You could see their behaviors that they were heat stressed a bit. But that was one aspect of my work, was being able to go from a body temperature in the field to say something about how well lizards were doing. Those are point estimates. At the same time, I was interested in thinking, well, can we go from beyond point estimates to some kind of an integrated measure of performance over time? And I started working with Monty Slatkin, distinguished evolutionary geneticist, then at the University of Chicago, and came up with a very simple model shown here that if you have a performance curve and you have a distribution of body temperatures, then you could do rate summation and get some kind of an average or overall measure. That's basically what you need. I'm going to borrow a graph from a beautiful paper that David wrote, colleagues, showing how you can get the integration. And obviously, if the body temperatures are clustered under the optimum, the fitness values are going to be clustered at high levels. So this model and variations on it has been used a lot over the years. And what I want to do is talk about some of the complications, some David has already raised, with the two components here, the distribution of body temperatures and also thermal performance curves. So first question, how do you measure temperature? Well, in herpetology, the classical way is to use a thermometer. It's a special quick reading thermometer that was conceived by Charles Bogert, for whom the Bogert effect is named. Martha knows very well. Developed in 1949. And here's an example of it being used. The late Carl Gans, Martha's won the Gans Award, and Phil Regal in Australia, with each holding a Schultheiss thermometer. They're about to take the temperature of this Tyrannosaurus rex. And I'm sure you've already guessed that the data that they gathered are a priori. They're smart enough not to take a posteriori data. Okay, so there's hundreds of thousands of body temperatures for lizards while they're active, but that's the problem. You can get it only when they're available and accessible. Another way is to use heat transfer models that Eric Riddell will be talking about later today. This is a project I'm doing with Michael Carney and Christopher Wild, where they're using heat transfer models to predict the body temperature of lizards in Australia in the 1960s, when Eric Pianka was working there. And you can see the red dots are the temperatures that Eric measured. The black dots are the predicted body temperatures. So these models work remarkably well to predict body temperatures a half a century ago. And that was even before that was 60 years ago. It was before I started working. Okay. Yet another way is to use telemetry. here are data from Frank Seebacher on an Australian monitor lizard. And one of the things that I find really intriguing about this is not a unimodal curve. It's nowhere near a normal curve. And what's happening is you have daytime temperatures and nighttime temperatures or cold weather temperatures, often leading to a bimodality. Okay. So having to do with risk, though, exposure, are these, as David pointed out, are these temperatures close to ones that are going to be hazardous or lethal? We know that the thermal environment is a dangerous place in many environments. I worked for several summers in southern Texas, Big Bend in July. And this is work from Bruce Grant and Art Dunham, where they're using biophysical models to measure what fraction of the ground surface area in Big Bend is above the lethal temperature of the lizards. And so early in the morning, it's great, but throughout most of the day, if a lizard put its foot out on those rocks, it's going to die for almost the entire day. Not surprisingly, the lizards are active only early in the morning and late in the afternoon. This was an early PowerPoint where I was able to use lizard scales for shading. It was amusing to me. Okay. Point is that the environment can be a dangerous place, but mobile animals, and that's an important thing, mobile animals like lizards can often use their behavior to evade those temperatures. This is a point that was raised by one of the key developers of evolutionary physiology, George Bartholomew, that behavioral avoidance is, for many organisms, a way to avoid stress. And this has been made in the context of climate warming by Jennifer Sunday, that behavior is going to be a key aspect. And that's something we need to incorporate as we develop thoughts about climate. So a key question, one David raised, is how hot are animals in nature? Are they close to their critical thermal maximum or not? And I worried about this when I was a postdoc and thought, well, why don't I find out? Go through the literature and look at the highest body temperature ever recorded. How close was that to the upper critical temperature? And this is what I found. And basically, the highest body temperature ever recorded is 6 degrees away from critical thermal maximum. The lizards and snakes weren't experiencing body temperatures that were close to the critical levels. Here's going back to Seebacher's data, where you have telemetry data. So these are much better fine-grained. Oops. Anyway, you can see that these lizards, even when we have telemetry data, so you're really good sampling, are not active at temperatures that are lethal. Okay, so, but we need more data like this to really get a picture on what animals are actually experiencing. And for terrestrial ones that can use their behavior, you can't just assume that the air temperature, operative temperatures are telling you what's going on. Another issue that was ignored when Monty and I were working, and it's ignored and intentionally ignored because it was complicated, is the fact that you can't just go from a temperature to performance without ignore the length of time that animals are exposed to that temperature. This is shown here in a paper from Joel Kingsolver and Art Woods, where this is a model where they're predicting growth rates of caterpillars. Okay. And at an instantaneous measure of growth, you get this curve. But if you hold them at those temperatures for longer and longer periods of time, the whole curve shifts, especially at high temperatures. This type of thing has been known for 100 years, but we've generally ignored it because it obviously complicates the analyses. But one question is an 80-hour, yeah, I think this is 80-hour exposure, is that ecologically relevant? I don't know for certain, but I don't think it is. But in any case, the point is that the body temperature distribution that animals experience will feed back on the performance curve. David pointed this out too. These are not independent things. How long do they spend at high temperatures? Here we've taken Seebacher's data, simply ask how long during the day the temperature rises, goes above some level, how long are they at or above that temperature? And you can see that when you get to really high body temperatures, they're not there very long. So how this plays out and how we develop our experiments to study the effects of duration of exposure is something I think is important that needs to be worked out. Another issue that Katie Marshall has emphasized with Brent Sinclair is not only a single exposure, but multiple exposures. And here I've taken data from Chris Wild on an Australian lizard. And you can see that sometimes you get heat waves, of course, and lizards can get hot multiple days in a row. What is the impact of multiple days of exposure to high temperature? We don't know. Certainly, very likely not good. It's going to affect the performance curve, but how it affects those curves is an issue. So I think just to sum up this part of the talk, you can ask, is knowing the distribution of body temperatures good enough? We know that time of exposure can be important, that repeated exposure to extreme temperatures can be important, but does that mess up our analyses enough? How sensitive are our models to those factors? I think that's an open question. And it may be that we need to switch and start thinking about time series of temperatures and incorporating frequency and time, duration. And also something I won't talk about is recovery from damage, heat damage, a really interesting but important area. Okay, let's switch to thermal performance curves themselves. There are some assumptions that go into this. That is, there is a thermal performance curve. We're going to ignore effects of ontogeny, sexual differences, condition differences. David mentioned these, and acclimation, recent thermal history. So we know that those are going to alter the shape and the position of the performance curve. But I think a real question is, by how much? Is it enough to really affect the outcome of our models? I think that's an open question. But first, let's look at the issue, is there a thermal performance curve? This is a plot I made back in 1982 for desert iguanas, and looking at various performance traits as a function of temperature. And several of them are pretty much on top of each other, except for digestion, which is key to energy balance, shuts off at a very high temperature. The distribution of body temperatures is there. So you can see the lizards are normally active at temperatures that they perform well. They're not running around very often at these low temperatures. So you might say, okay, that's performance. What about fitness? That's hard. In 2001, David Berrigan and I did an analysis where we took life table data for maybe 50 or 60 species of insects and invertebrates where people had measured life tables at multiple temperatures. So we could calculate two classical measures of fitness, little r, the intrinsic rate of population growth, or r naught, net reproductive value. And here I picked an extreme case. But before we even analyzed the data, we knew that this is going to happen because little r is sensitive to development time. But these are radically different curves. So if you're putting that into a model, which curve do you use? I mean, obviously, you have to tune it to the biology of the species that you're studying. And there are other problems with fitness curves as well. But it's a challenge. Okay, there's another aspect here about thermal performance curves, and that is that we're ignoring the environment. As David mentioned, there's competitors or predators that can influence the ability of animals to find environments that are suitable. But there's another factor that I want to discuss, and that has to do with food. And that's been prompted by a series of papers in the last 10 or 15 years showing declines in insect populations And this is a paper from Germany looking at insect biomass. I'll do the old-fashioned way. Insect biomass over time. And this is on a log scale, but there's basically a 75% drop in the biomass of insects. Okay, that means for birds and lizards and frogs that are running around trying to find insects for food, their ability to forage is going to be reduced. They're probably getting less food. Okay, and then that led me to a paper that I read, actually not sure when, but I think I was probably a postdoc when I discovered this paper. Fortunately, I had not discovered it when I was a grad student because Brett, this fish physiologist in Canada, had this plot looking at temperature and growth rate, and I'm going to reproduce it here so it's easier to see. So he's manipulating temperature and food level and looking at the effect on growth rates of salmon. And anything above that horizontal line, the fish are growing. Anything below the line, they're starving because they're not being fed and they're losing mass. Okay, you'll notice that when you provide them excess food, they grow a lot, no surprise. And as you start giving them less and less food, they're not able to grow as fast at any temperature. But what's really wild is that the temperature at which growth, positive growth is possible gets less and less. In effect, these fish are less heat tolerant the less food you give them. Moreover, the optimal temperatures are switching to lower and lower levels. So the curve isn't just that they're dropping, it's shifting like that. And when I saw this, I was horrified because I liked my idea about thermal performance curves, but suddenly I realized, hey, there's not one, there's an infinite number of performance curves. So initially that was very upsetting. It took me a couple of years to get over it. But I was intrigued by this plot, and I kept coming back and wondering, what's going on? And that led to a simple model that Reed Hainsworth and Blair Wolf had done a few years before it, but we both converged independently. The idea that growth is the difference between energy in and energy out. So metabolic rate scales somewhat exponentially, and the maximum growth is when the two curves are maximally different. And if you reduce the amount of food, you get a reduction in growth rate and a shift of the curves to a lower temperature, basically Brett's curve. Okay, that was 1982. I didn't think much more about Brett. I mean, I taught it every time I taught physiology because I just loved this paper. But until climate warming started coming along and these papers on food levels started coming out, they started putting these two things together. And I want to call attention to a really wonderful paper that Rajul Thomas and colleagues did. It's a very different model, but it's a population model rather than energetic model. But they're showing the same thing as you lower the nutrient availability to phytoplankton, getting Brett-type curves. So if you're a behavioral biologist and you look at that and say, well, if that's the case and the food is dropping, what should an ectotherm do? Move to a lower temperature because it's energetically advantageous. And this is the case. This is a study from Fiona Clissold and colleagues in Australia, where they put locusts in a thermal gradient. And this is a heat map, so you're looking down. And so most of the locusts are clustered between 36 and 39. Give them second food, they stay there. But if another group, you take food away from them, notice that they move to a lower temperature here. So they're doing exactly what you'd predict based on energetics. So when climate warming became an issue and these food papers were coming out, I thought, time to revisit Brett. And had the presence of mind to work with Joel Kingsolver, who's an old friend and done a lot of work together, and published a paper in 2019. I'm not going to do the math, I'm going to do a graphical presentation of the idea. But the thing is, first of all, imagine an environment. You can go out and use biophysical techniques to estimate the coldest temperature and the warmest temperature in the environment. So an animal that comes out is going to be somewhere in that range. And you can do that with biophysical techniques, or as Martha has used, copper models, which you can deploy, which work very well also. Now you specify a range of body temperatures that are acceptable for activity, and that allows you to predict the times of day that these animals can be active. And in this case, they can be active pretty much all day long. Now let's see what happens when we impose climate warming. So those operative temperature curves shift up because it's a hotter environment. And notice the consequence is that there's now a shorter activity time during the day. It's a point developed by Barry Sonervo and colleagues back in 2010. Now, if they have less time to forage, that probably means they're getting less food. And if the food level of the environment is there's less food out there, that's even more reason that they're going to be getting less food. So what happens then? Well, they should shift to a lower temperature. And that's shown here. And the effect is to reduce the activity time even further and reduce the amount of food that they're getting. So in effect, you end up with what Joel and I called metabolic meltdown. That is, basically, you're going from a bad situation to even worse when you combine a reduction in food. Okay, so where does this leave us? I think this approach is useful, but there are a lot of complications that make life challenging, both with the distribution of body temperatures and the idea of thermal performance curves. And I think we really are only at the beginning of understanding the effects of acute and chronic damage, exposure to extreme cold or extreme heat, and trying to incorporate those into the models, something that's important to do. And I think the bottom line is that this field is increasingly complex. I mean, we do a lot of this when we were starting, but just chose to ignore it for obvious reasons. But whenever I think about complexity, I'm always intrigued by a comment that Richard Levins wrote many, many years ago, that if you have a complex system like thermal biology, you can just try to describe it mathematically in really complex models that probably only the person describing it, doing it, can understand. And the real trick here is to try to simplify in the models in the way that are realistic, but still capture the estimate, the essence of the problem. And I think that's a real challenge, and it's going to be exciting to see how people are able to handle that. So it's been 50 years. There must be an arithmetic error somewhere. But anyway, it's been a lot of fun working in this field. I've had the opportunity to work with amazing biologists and friends, and I'm really looking forward to the rest of this week with learning some new things from many of the people in this audience. Thank you.
Great. Well, we have some time for questions, so I'll let you.
Sure. Sure.
So I have like two thoughts. And I think you're already going to disagree with this.
You're right.
We're moving into this age of omics, right? And
Yep.
you almost want to apply, like, is there a thermal performance curve omics? But the end of your message was kind of saying, let's not do that. Which I definitely agree with. The other thing that I wanted to comment on too, which I thought was really interesting about Brett's curves, was the starved individual. Moved from convex to cave curve, but there's clearly some sort of acclimation happening there where even as you move up the same to warmer temperatures, growth rate doesn't decline more. You still don't have, the growth rate was equally low at 20 degrees as it was at 30 degrees.
Yeah, that's interesting.
Even at warmer temperatures, growth rates
Should go down.
so there's some sort of, is it acclimation in that case? And do you think that those basically what I'm trying to get at is, do you think that acclimation is playing sort of a stronger role in when these fish are starved, and potentially less of a role in this type of experiment where they have access to food? And I'm just curious if acclimation could particularly buffer some of these conditions where they have access to less food.
Well, anyone who's attempted to lose weight knows that initially you lose mass, but then your metabolic rate drops and it's much, much harder. So that might be going on. It's also possible, man, I hadn't noticed that, so thanks for putting it out. It's also possible that these fishes were in tanks with multiple tanks. And I'm wondering whether they're eating poop, eating each other. And there's some potential artifacts that could be involved there. I know in working with flies that we had flies scavenging off other flies in starvation experiments. So I don't know, but it's, and I don't know of anyone who's followed this up with Brits, with those salmon, but it'd be interesting to do. Katie.
So this also is kind of playing off that last comment that you made. The most accurate map of the world is the world itself. That's not necessarily the best map, right? The best map is what, is really defined by what you want to use it for. So, sometimes you need a roadmap for thriving. Sometimes you need like a really zoomed out map for understanding what's happening on broad scales. So I guess when we think about this, are there sort of tools or ways that we can think about what it is we're trying to do that can maybe capture some of this complexity in the places that we need it, but not overwhelm us with complexity?
I don't know. I think the first thought is you can develop a whole series of individual models and compile them and change the scale, as David was mentioning, both temporal and spatial. I haven't done that, and we've looked at geographic patterns, but I think that's an interesting question. I mean, my initial instinct is to work from a single species and then add, but that's just because of my background. I haven't thought about it the other way. Can you go from a community level down? I suspect there are insights that I would miss, but yeah, I don't know. Please do the work and tell me the results.
Yeah, fair enough.
Martha.
I have a question related to metabolic meltdown. So the behavioral shift to cooler temperatures relies upon the behavioral capacity to do that, right? Do you think there could be a difference in the signatures, therefore, of metabolic meltdown between thermal regulators and thermal conformers? In other words, are thermal conformers, are they capable of seeking cooler temperatures within their environment, or are they truly passive with respect to their thermal environment?
Oh, never thought of that. Thank you. Based on thinking about Anolis cristatalis or Anolis in the Caribbean, where there are a lot of thermoconformers, the only opportunity they would have is to restrict activity time. And because, I mean, these guys spend 24 hours a day outside, and the heterogeneity in operative temperatures in that forest is trivial. I mean, it doesn't exist. So there's no opportunity for microhabitat selection, or very little. So I don't think they have an option other than restricting time of activity. They can crawl down a burrow or get under the bark of a tree or something like that. Whereas an open habitat animal can change time of activity, change time spent in the sun, amount of time spent basking. There are a whole series of behavioral options that are available to those animals. And just realize, I don't think anyone has ever gone to the field in the middle of the day and looked to see where, well, middle of the day, they're all in the shade. Maybe, but in the morning, find out lizards that are basking and the ones that are in the shade then, do they have differences in the amount of food? I mean, decades ago, yeah, in fact, as the lead paper in ecology for this millennium. Eric Piak and I had a paper on feeding in lizards, and we showed that most lizards in the field, the vast majority, have food in their stomachs. So for them, a bread effect probably is not really critical. But that can't be true for many other animals, that being unfed is going to be a common thing of life. But for lizards, at least in the data we had for multiple deserts in the tropics, they're fed. So I think it's less of a problem for them. Better.
I have one more. It's related to what you were just saying. So I guess I was now taking that like kind of further. Like if metabolic meltdown, restricted time for feeding, is there any? And you're suggesting maybe that they're not actually food limited as of yet. And I was wondering if if with warming in some situations their food resources become more prolific, or their handling time, whatever is limiting them, it's getting faster, then maybe there could be some trade-off, but it sounds like they're not actually for the most of it.
Yeah. Although I just remember back to a paper that Paul Lick did back in the 1960s. Paul was working in Puerto Rico, and he took Anolis cristatellus and he went around and fed some of the individuals and not fed the others. This is on the campus of the University of Puerto Rico, so it's not an ideal place to do the project because they probably spray for insects there. But he showed that if you feed them mealworms, they grow. They put on fat, so they were not operating at full capacity in nature. I don't know if anyone has followed that up, but it was a very clever idea and easy to do. Actually, Gary Ferguson has done some work with UTA feeding them, looking at life history effects of feeding. But yeah, it's such an obvious thing to do. And I guess you could take food away and see what do they do in the field. I don't know anyone who's done that. That's a little trickier to take food away from an animal. But could be done. Yes?
I'd like to follow up on the last two questions, where you get to the question of ecology of energy storage utilization, possible effects of its microbiome, its effectiveness, mobilize, helping research from all perspectives.
This is not something I've worked on, so I can't say. I do know that there was an amazing study done on aphids. For some reason, I'm drawing a blank. University of Texas, what's your name? Norwell. She took aphids and was able to show that these aphids have a parasite, Buchnera, and one base pair mutation in the parasite, it's an endoparasite, affects the heat tolerance of the aphids. That's the most upsetting thing I've ever seen. I think that's why I was spacing out on the actual paper and the person did it. It was, when I read that, I thought, that's a frog. Okay, I think I understand something about ectotherms. They're individuals, I've studied them my whole life, and suddenly realized it doesn't have its own physiology. I mean, if I were starting again as a graduate student, that's what I'd be working on, because it completely upsets my entire life's work. And it's very traumatizing. Anyway, I'm embarrassed that I'm drawing a blank on the person who did this work, but it's a fabulous study to show major effects on the heat tolerance of an organism. That's obviously going to apply to everything. Just one base pair mutation in the Buchnera. That's really upsetting. So there's a lot of options, but yeah, I wish I knew the answer to that. It's very likely, certainly worth exploring just to see what happens. There are people working on microbiomes in lizards. I'm just not, I'm not involved.
All right, thank you very much, Fred. We'll keep things moving.
Okay.
It's my pleasure to introduce Dr. Katie Marshall from the University of British Columbia to join us today. I have to say, most of us tend to think about the warm end of the thermal performance curve, and Katie very much then is our anchor that keeps us reminded that there are things going on the other end of the spectrum as well. So I'm really excited to learn more about what's happening on the other side of climate change, and Katie's work has been extremely influential in looking at how cold adaptations, cold tolerances play out in insect populations, and also thinking about what happens as the cold is disappearing from these systems and guiding us and keeping us honest, I think, in the thermal performance community, that we can't neglect what's happening on the cold side. So with that, I'll pass it over to you. So thank you very much for joining us today.
Thank you for that. I'm going to get out of the glare of the light. I like to be not behind the podium because I'm quite short and I kind of hide behind it. So can everyone in the back hear me okay? Okay, awesome. First, thank you, Martha and David, for that wonderful introduction, for bringing me here, for bringing my students here, who I saw just made it after traveling all night. And before I get started, I also want to acknowledge that the work that I do takes place on the traditional, ancestral, and unceded territory of the Musqueam people. And if you don't know much about the Coast Salish people in Canada's Pacific Southwest, and we're going to be looking at things from the Canadian perspective this morning, I definitely recommend looking at some of their history and their ongoing cultures. It's really, really amazing stuff. And as David introduced, I think we are so used to thinking about climate change from the heat perspective. Summers are getting warmer. And so what I've plotted here is we're looking straight down from the North Pole onto the northern hemisphere of the world. And you can see, yes, we have seen significant climate change in the summer months. But what happens when we look at the winter months specifically? All of a sudden, you can see that these patterns become way more intense. And this is true no matter sort of how you slice it. Winters are changing much, much more rapidly than summers are. And in addition to this, what you can also see is that we often are getting these latitudinal trends in the degree of that change. So the further forward you go, the faster that change is happening. And so as a result, species ranges are moving. And I know Jen is going to give you an amazing talk about some of the complexities of this, but I'm not going to steal her talk. But one of the things we did do is look at across, particularly tropical ecosystems are moving into the temperate zone. And with that, species are moving quite a lot. But the degree of that moving is actually really species specific. And in some cases are actually moving craterward. It's really complicated. So the way that our lab likes to address some of these questions, and I think Ray really nicely introduced, is that we think that understanding mechanism can help us make better predictions. And the example I like to give is that we take a correlation of this grasshopper jumping over time, and we project that into the future. A strictly correlative prediction is going to tell us that that grasshopper is going to keep going up and up, maybe hit the stratosphere, maybe make it to the moon, right? It's not very realistic. But if we know something about the air resistance due to the force due to air resistance, we know something about the force due to gravity, we can make a much more accurate, potentially, prediction. And this was not my idea. This is a great paper published 8 years ago now. I highly recommend you take a look at it. So let's think about winter. This is what winter looks like in Canada, mostly. And the thing about it is that there's two key issues for an overwintering organism. Winter's cold and it's unproductive. There's often not a lot of food around. And so organisms respond, I almost think of it as like two sides of the same coin. On one hand, we see downregulation of metabolism. We see often an arrest in development, and often a cessation of feeding. There's no food around, so you might as well batten down the hatches, do the best you can with what you've got. So we often call this dormancy, and this occurs across a huge range of species, vertebrates, invertebrates, plants, phytoplankton even undergoes some dormancies. So this is a really, really universal response. On the other hand, we often see upregulation of stress tolerance, cold stress tolerance. So often cryoprotectants of various sorts, sometimes alternative isoforms of proteins are expressed that allow them to maintain activity or at least not die of cold stress. So we have two sides of the same coin. So let's think about this in the context of warming winters. Oh, and I should mention actually, this overwintering time, depending on the organism, can be 80% of the total life cycle. So I think we can't just ignore that this is like they go away somewhere, they hide for the winter, and nothing interesting happens. This is the majority of many, many ectotherms' lifespans. Okay, so how would winter climate change potentially affect these? Well, warmer winters might actually cause greater energy drain. If you're not able to feed, and so all metabolism is coming from the reserves that you have at the beginning of winter, the higher the temperature, the faster you're going to run through those energy reserves, right? So metabolic rate increases with temperature. But on the other hand, the other side of the coin is warmer winters might cause less cold stress. So we can see both benefits and drawbacks to a warmer winter, depending on the organism. And so what I'm going to be doing in this talk is talking a little bit about my own lab's work and then putting it in the context of what we know broadly across lots of organisms. But there are tons of unknowns about all of this. And so this is sort of hypothetical. I'll show you what data we've got, and I'll highlight some key unknowns about this whole thing. So given the symposium and given just who I finished talking with, let's take a look at a thermal performance curve. And often, and I always look for this in thermal performance curve papers, often the bottom end of this curve, there we go, is poorly drawn out. And part of that is technical limitations. It can be hard to cool down equipment. It also can be hard to really figure out where CT min is, because what you see is this kind of like long, slow, decline here. And so if we really believe in such a thing as a thermal fitness curve, with all of the caveats that Ray introduced, we really need to look at this a little more carefully, I think. We want to somehow link performance and fitness, or at least we want to understand what that fitness curve could look like. And so the way we often think about it in the cold tolerance world is really that organisms pass through a series of important physiological thresholds in the cold. And so let's imagine we take this little lady beetle, we cool her down over time. I'm an invertebrate biologist, so there's going to be a lot of bugs in this talk. My apologies. So the first thing that's going to happen, or at least the easiest to measure thing, is a CT min. We call it chill coma. There is, I think, broadly an issue with integration of many disciplines, but we would call that chill coma. And so this is a reversible loss of neuromuscular function. But if we're thinking about this in the context of fitness for an animal that's in some kind of dormancy, it's found a den or a burrow, this is probably not really a fitness problem, and you can't measure it anyway. Like if we've got a hibernating ground squirrel, as I've got here, what's a CT min really for it, right? It's already not moving. So how do we measure a locomotor CT min? We really can't. So once organisms spend enough time in the cold, they start accumulating some chilling injury. And as Ray mentioned, this is time dependent, time and temperature together. Same thing as you would see on the heat end. And this is poorly characterized for a lot of organisms, but this will generally kill most organisms we think about. In the cold world, we call this chill susceptibility, and most ectotherms are going to be chill susceptible. However, there's further thresholds. So at some point, you risk freezing. And now this temperature can be really suppressed. We're going to look at a few examples of that today. And a lot of pest insect species actually fall into this category. They suppress their freezing point. They can't survive freezing, but they can go down well into sub-zero temperatures. And so this is the emerald ash borer. This is an invasive insect here found in eastern North America. I'll show you some data on this. They can really suppress that supercooling point. And then finally, we have our freeze-tolerant species. We work on a few of these in the lab. We're not going to spend much time talking about it today, but if you've ever seen a woolly bear caterpillar, I don't know if any of you do, that cross the roads in the middle of winter, these are freeze-tolerant. They can survive internal ice formation and not die, which is pretty amazing. They accumulate freezing injuries at some point. I've mentioned a piece of jargon. I'm very sorry. We're going to be stuck with it. This is the temperature at which an organism freezes. It's called a supercooling point because that's where supercooling ends. And we get a little bit of heat release. We can measure this easily in the lab. We do this all the time. And so this is going to come up a lot when we're talking about these lower thermal limits. So these are directly fitness limits, right? Where mortality occurs. Okay. Now the thing about lower thermal limits, in particular relative to upper thermal limits, is they can be very, very plastic. This is the supercooling point, so the temperature at which emerald ash borer freezes and dies, and it's through the winter, and you can see that's a 10-degree spread of temperatures. Like, it's really plastic, right? And if we were to measure them in the summer, it would be even higher. So they really are able to be quite plastic. This plasticity itself can also evolve. So when I was in grad school, I worked with Casper, and we looked a little bit at plasticity of Drosophila. So this is a cross of Drosophila phylogeny. I'm sorry, a little pun there, just couldn't make it. I couldn't, yeah. And so what he did is he measured survival from a cold shock in Drosophila, and that's in the black bars here. And then he also gave them just a 2-hour pre-treatment, so a little bit of a cool temperature, not enough to kill them, and then measured that survival at that same temperature again. And so what you can see is that we had, and these were all, I would say, mentioned, are all reared in the same lab environment. What you can see is that some species, like Drosophila takahashi, are really able to increase that cold tolerance. And others, like Drosophila pseudo-obscura, are really not. So we see differences in plasticity even across, relatively closely related species. So this is really fascinating because just 2 hours of cold exposure actually induce significant cold hardiness. So this tells us that plasticity itself evolves along with direct hardiness itself. Okay. So can we use some of these mechanisms of overwintering, these two sides of the same coin, cold tolerance and that plasticity, dormancy and that plasticity, to better predict range expansion under climate change? And this has been one of the topics we've been thinking a lot about in my lab group. So I'm going to introduce you to one of my favorite species, and let's just see if I can make that video play. Oh, no, there we go. Okay. There we go. So this is not sawdust. This is not dirt. This is moss. This is the eastern spruce budworm in its outbreaking phase. This is one of the major pest species of the boreal forest in North America. And you can see it is just everywhere. And just to give you a sense of what the outbreaks of spruce budworm look like, this is over the last decade or so. This is the number of hectares of the boreal forest that is impacted by spruce budworm. And this is 600,000 hectares. I don't know if that's a unit we use here in America, but it's about 4.5 times the area of Connecticut, just to give you a sense. So it's a lot. It's also, I think, like 14 million football fields, I think. So, yeah. And the reason this is a problem is that spruce budworm is a defoliator. It eats the buds of growing trees and eventually kills them. And so as a result of this action, we're actually seeing the spruce budworm turning the boreal forest into a net carbon source rather than a carbon sink. If we think about the scale of this, obviously a big problem. Oh, well, keep hitting those buttons. And what we know from past modeling work is that warming winters are actually modifying these outbreak patterns, allowing the budworm to survive more of the winter. They have a limit for how cold they can handle. And if we take a look at the life cycle of the spruce budworm, you can see the majority of their lifespan is spent in blue in that overwintering stage and 1 generation a year. And so we can imagine that the things that happen during this long overwintering stage are really important for predicting the overall survival and abundance of this species. I just want to show you, this is the range of the eastern spruce budworm all the way from Alaska to Newfoundland. So it is really, really widespread. There's also this really cool disjunct population down here. We're just starting to work on it. I'm super excited to see what we find. So one of the things I love to do is you go back into the literature, and this is not even that long ago, but people were doing really amazing work, and sometimes we just don't think about it. And this is one of those graphs that always blows my mind a little bit, because what they did is they took spruce budworm, they overwintered them in the lab, and then they took the same strain and they also put some of them out in the field. And what you can see is that that lower lethal temperature is really not doing much in the lab maintained population, but in the field acclimatized population, you get this really impressive suppression of that lower lethal temperature. About 8 degrees, kind of like the emerald ash borer, it's a really big deal. And probably one of the drivers of that is this accumulation of glycerol, which is an important crowd protectant for the species. And you see the same pattern. The lab maintained critters don't accumulate much glycerol, but the field acclimatized ones do. And so when I was in grad school and I came across this data, I was like, man, we're measuring cold hardiness like all wrong, right? Like we're really somehow missing some important driver variation in cold hardiness. And it seems to me, if we think about the lab versus the field, what are the things that are different? Well, the field has thermal variation, right? So I'm going to show you one of those graphs that you like spend 2 years putting together, and it looks like this. It's like 3 bars. And what I found is that fluctuating temperatures, no matter the temperature I fluctuated them at, so it really acted as almost like a threshold effect, causes accumulation of glycerol, quite a lot. We got about 4 times as much glycerol as we did in the control population. You could really induce this kind of plasticity. The problem with what I spent years of my life doing is it was all based on one colony that had been reared in culture for about 60 years, and so we don't know if this actually, reflects what's going on in the real world. So I was really lucky as I started my first faculty position. Amanda Rowe and Ashlyn Wardlaw from the Canadian Forest Service were establishing budworm cultures from across the range, so all the way up 60 degrees latitude in Nunavut, Northwest Territories. And Skye Bedison, my first graduate student in the lab, she measured that supercooling point in different populations under basal fluctuating conditions. And what she showed, and also glycerol, what she showed really nicely is not only are there these evolved basal differences in cold hardiness, but also the plasticity itself has evolved. So we can see that our more northern populations are also much more plastic. And one of the things that's really interesting is some of the work on population differentiation of this species has shown that there's actually a SNP in glycerol-3 phosphate dehydrogenase, so that's part of the glycerol accumulation pathway, actually differentiates these populations, suggesting it's under selection, which is cool. And I'm not the only one to think a little bit about wintering temperatures and outbreaking insects. Warmer winters have led to greater outbreaks in the mountain pine beetle, as well as a range expansion of mosquitoes that carry important diseases like dengue and malaria. And so this is something that I think many, many people are thinking about when it comes to pest species. So if we think about some key unknowns for predicting the effects of warming winters, first, how plastic is cold tolerance really? Are we actually measuring the full extent of that plasticity? And secondly, how evolvable is this plasticity? What selection has there been for it? Now, what about the other side of this coin? What about dormancy? Well, dormancy is actually a really common response to winter. We see it across a wide range of species. And in dormancy, you can almost think of 2 important axes. How deep that dormancy is. Is it a shallow dormancy, a really deep one with a lot of suppression? And how programmed is it? So programming is when we've got this central nervous system driver, this endogenous timekeeping that really, photoperiod triggers the onset of dormancy and they are stuck. Like if you keep them in warm temperatures, they still become dormant. On the other hand, we can think of species where dormancy is driven by a direct response to the environment. So it's just, they experience some cold, they go into some kind of quiescence. And if you plot different species, you can sort of see them occurring in all different sides of these 2 axes. So we have vertebrates, we have invertebrates, we can really plot just about anything there based on how programmed it is and how deep that dormancy is. And metabolic rate for organisms that undergo a deep dormancy is really, really suppressed. This is a flesh fly, Crassipulpus, and here you've got metabolic rate on the y-axis. It's all at the same temperature, but you can see that deep, deep metabolic suppression. And this is really important when we think about this from kind of a thermal performance curve perspective, because if you're not feeding and your metabolic rate is high, that's going to be a problem. So one of the ideas I'm just going to throw out there, I don't know the answer if this is true, but we might actually think that a thermal performance curve might be mirrored in the winter. Or if we think about this in terms of a thermal fitness curve, these abrupt lower thermal limits, but at high temperatures, maybe energetic drain is a problem. So let's take a look at what that might look like. Dormancy begins early when temperatures and energetic costs are high. Remember, for many organisms, this is programmed. So this is showing onset of dormancy relative to onset of winter across a wide range of insect species. And I've plotted the one-to-one curve here, so you can see they're really entering dormancy almost a month ahead of when winter conditions might happen. And if we think about this in terms of metabolic rate, so this is some work that Caroline Williams and I did when we were both in grad school. We've got CO2 production as a function of temperature. And here we've got the temperatures in the environment of this organism. They're from actually Oregon, so not that far south from a Canadian perspective. And in purple, I've highlighted the periods in which they are in diapause, when they're in that dormancy. And in particular, you can see this early period, temperatures are hitting almost 30 degrees Celsius. They're dormant, they're pupating, they cannot feed, they can't escape, right? They are just stuck where they are, and that's it. That's all they have to deal with. So why do organisms do this? It makes no sense. If dormancy is so costly, why do organisms do it so early, right? Well, one thing to understand is that dormancy and diapause are a series of programmed responses for the programmed species. And I've just listed some of them here. I can't get into it in great detail, but maybe this initiating phase actually requires enough heat to be able to undergo these effects. Maybe there's some developmental effort that's important here. So we decided to explore this in the eastern spruce budworm. And when you've got a pest species, you can get like a lot of them. And we do these experiments with like 50 experimental groups. It's a little bit wild. But in this case, what Ashlyn and Amanda did is they took these animals, they kept them in the fall at 4 different temperatures, so 10 up to 25 degrees, and for different lengths of time. So we explored that time axis too, between 1 and 10 weeks. They then overwintered them, and we brought them out and looked at how their survival was. Now, Skye looked at the energetics here. So we look at the amount of glycogen, which is our main overwintering energy reserve. And as you might expect, a warm, long fall leads to a significant decline in energy reserves. And that makes total sense. Cooler short falls, you have a lot more energy. Makes total sense. And if we look at this like partway, midway through the winter, we see the same pattern. They're really not changing. They're not kind of catching up. It's really that energy loss in the fall that's important. When we look at survival, high temperatures for a long period of time lead to very little overwintering survival. Makes total sense, as you might expect. Well, we get some weird things at the low temperature. So we have this increase in mortality with a long warm fall, but a short cool fall, look at that. They don't survive at all. It's just like, what is going on there? So on the x-axis, we have time at the temperature, and the y-axis, survival. You can see that like you need enough time in the cold to actually have high survival. So glycogen depletion makes sense, right? We kind of plot this as a heat map. On the x, we have temperature. On the y, we have that time at temperature. And as you might expect, the more time and heat, the less energy reserves they have for overwintering. But survival does something really different. So what we have is low survival with the warm long fall, but low survival with a short cool fall too. If anything, you see higher survival along the diagonal here. So one way to think about this, if I can get this to go, we've got slower development in the cool, quicker development in the warm. So maybe what's happening is that there's going to be some kind of necessary, you need enough heat to actually complete all those developmental processes to successfully overwinter. We're not sure. We actually have very little understanding other than, there's a photoperiodic trigger or a life cycle trigger that initiates dormancy, but we actually don't know the physiological processes really at all. We've been doing some RNA sequencing to try and get a little bit at this. We see things like carbohydrate metabolism, cell signaling, ion transport being differentially expressed between diapausing and non-diapausing strains. We also have completed genomes for diapausing and non-diapausing strains. That'll be data that's coming out in the next little while. I'm very excited to talk about it. But really, we see this massive genetic gene expression change, but very little genetic change in sort of this diapause. So really interesting stuff. Okay. So to recap, fluctuating temperatures induce additional cold hardiness. This varies regionally, And that diapause onset in the fall is also strongly, it also strongly impacts overwintering success. So can we use this to actually understand how outbreaks might change on a landscape? Well, it turns out with spruce flyworm, you can actually get really good data on population density. So every year, the Canadian government sends out airplanes to actually monitor the amount of defoliation that has happened across the landscape. So this is New Brunswick, Quebec, to give you a sense, Gulf of St. Lawrence, orient you a little bit. And this is real presence absence data and actually a measure of population size in every grid cell. It's really amazing. There's very few species we get this kind of fine scale data for. And so Emily Black, she's actually here somewhere. Oh, yeah, there she is. She's a PhD student in the lab, and she's been building species distribution models that take some of these effects into account. So here she has, in this random forest model of spruce budworm defoliation, she's built in time of year, she's built in the number of thermal fluctuations across various thresholds. And what I want you to be able to see here is that these fluctuation variables really are important for predicting spruce budworm population density. What's also interesting, and what I've highlighted here, is what happens in the fall is also really important. So we see these same things that we are able to measure in the lab popping out in these models that we're building of spruce budworm defoliation risk. Now, what happens when she actually predicts into the future? What does that look like? So this is a model of what defoliation might look like in 2050 without thermal variation. And if you include those thermal variation variables, there's a couple of things you can see. First, we get a lot higher defoliation, and that the location of that defoliation really varies as well, especially here in the northwest of this particular part of the range. So we know that our models are actually really sensitive to this fluctuation variable. Okay, so just to recap, we've got fluctuating temperatures, we have fall diapause conditions are all important, and it seems like including these effects in at least species distribution models here, it improves accuracy and does really impact the types of predictions we make under climate change. Now, is this the only organism that maybe the fall matters for? Or that dormancy matters for? No, there's been some really interesting work on that population declines in butterflies. And what they found in areas of higher fall warming, that butterfly populations are actually declining more. And it makes sense that dormancy is this really vulnerable time because you're not able to behaviorally thermoregulate, you're not able to feed, replenish energy reserves. And so warming could be actually really detrimental during this stage, especially in the fall where things are pretty warm already. Okay, so a third key unknown, I think. What are the geographic and taxonomic patterns in dormancy, length, and occurrence? We actually don't have a good handle on this. I was chatting with Ray before the talks. We actually know very little about brumation and lizards, which is their form of dormancy. Probably insects are the best, the most taxonomically sort of diverse range of dormancy responses we've got. But this is not, just in insects. This is across many, many different taxa. And secondly, what does a winter thermal fitness curve look like? Can you build one? And how does it integrate with the rest of the life cycle? And I think that is also something that we really just don't know. So I'm going to leave you with one last thought, and this is just because I think it's kind of an interesting idea, and it's been something I've been thinking about lately. One of the interesting things when we think on a global scale about temperature is we're sort of used to thinking about the equator being really warm and the poles being really cold. The other thing we get is this really strong gradient in seasonality, right? So the further poleward we go, the more seasonal climates are, particularly in the northern hemisphere. So if you are an organism that's moving poleward due to climate change, due to some kind of climate warming, is there the possibility that you might actually, as you're like following maybe a summer isotherm further poleward, all of a sudden you reach an area with greater seasonality? Can these warm adapted species maybe actually get nailed with cold. You know, they've just made it too far north. And I don't know if you've ever seen, there's great YouTube videos of iguanas in Florida, these invasive iguanas falling out of trees when they get cold enough. Highly recommend it. This will be a more sobering, unfortunate example. So sea turtles can become cold stunned. And what that is they get so cold that they're unable to swim, and they often wash ashore, and this can kill them if they're not warmed up in a gentle, sustainable way. And what's interesting is off of Cape Cod, the number of cold stunning events has actually increased significantly over the last several decades. And the causes for this are not totally understood, but the thought is maybe shifting currents. They're moving a little bit further poleward, but they're just not able to handle that increased seasonality in the cold. We've also done a little bit of work on this. I worked with Shahar Chaikin, and what we were interested in is cross-marine fish. The species that are moving poleward the fastest, how are their populations doing? And so he used the bio-shifts and bio-time data sets. And what he found is that the fastest shifting species are actually the ones whose populations are declining the most. And on one hand, you might think, oh, maybe they're just like extremely vulnerable and they've got like very few, options, right? But it turns out that it's actually the ones that are at the poleward, the cold range edge, whose populations are declining the most. And so is this a situation, again, where they've moved poleward and are just like unable to handle winter when it hits? It's almost like a thermal trap. So finally, I want to leave one last sort of key note, which I think is interesting, is what is happening to seasonality isotherms, and how do we sort of think about not just warming, but kind of across the entire life cycle of species. So species ranges are moving, and I'm hoping that I'm setting Jen up well here for her talk. And on one hand, you can imagine reduced cold stress is allowing species to move forward. Maybe seasonality is something that's preventing them from moving as far forward as they could. How does warming winters actually play into all of this? So I think understanding winter and dormancy can maybe help us understand this a little bit better. I'd like to thank my funders, my lab group, and I think I've got a few minutes for questions.
Just try.
Sure.
You had a map early that showed warmer colors, more change of some sort, I guess more warming, and cooler colors showing less negative.
Yeah.
The cooler was all over Asia.
Yeah.
Which was different than
Yeah.
the final maps.
Yeah. I will.
So that's noticing a difference. I'm not sure what the question should be.
Yeah, no, it's super interesting. This has actually been something I've been thinking about too in the context of where I work in Vancouver. We are getting there we go. Here's the map you were talking about, right? Yeah, so one of the really interesting things is we're actually getting cold snaps in that, and anyone who's lived through a polar vortex also knows this, right? In Vancouver, of the 5 coldest temperatures on record in Vancouver, I think 4 of them have happened in the last decade, which is like mind-blowing, right? And so what we think is happening broadly with winter is that it's not just a matter of warming, it's actually a shifting of thermal masses. And so I think what's happening is this kind of shift of thermal mass to North America, which is causing cooling in Asia. So even with climate change, I think we need to think about cold. So, yeah.
I guess are Russia. Are there any studies that are talking about the things you talk about, but studying them in Western Russia?
Not much as far as I'm aware, but yeah, we did work with someone from Yakutsk, and she was working on these because it's extremely seasonal as well, because you've got this big sort of landmass. And these like butterflies that could handle like minus 45 degrees Celsius, right? They survived Siberia, so this is not surprising. But yeah, there's just, I would say most of the work that's been done is really focused on Western Europe, North America, and obviously we're missing huge pieces of the puzzle. So yeah, totally. Yeah.
I'm
I'm really interested in your idea about an inverted thermal performance.
Oh, yeah. Oh, I was very excited about that.
So we'll talk more
Yeah.
about that.
Yeah.
But what do you think about the sort of trade-off between, how much energy is being lost due to warmer periods during dormancy, but potentially having shorter dormancy periods? Are
Yeah.
those two things compensating for one another?
So that is a very much an open question. I would say the onset of dormancy, and I'm just going to show you this little thing again. The onset of dormancy tends to be, at least for some species, most species that I think about, is programmed. So it's driven by a photoperiodic cue. Those are not shifting in the same way that temperature cues are. Now, whether they're able to terminate, so we think termination is going to be more driven by accumulation of degree days, that that programming kind of fades out through the course of dormancy. We don't know if that's broadly true or if that's just an insect thing. If they are able to come out, maybe there's like a bit of a trade-off with, that they can have a shorter overwintering period, but we don't know. Bill Bradshaw has also shown that there's been evolution in the photoperiodic response that does, over evolutionary time, or at least over the last couple of decades, has led to onset of dormancy shifting with climate change as well. So there's a whole phenology section that I would love to cover, but didn't have time. So yeah, totally. Yeah. Was that what you were going to say? Okay. Yeah.
So I'm intrigued about sort of the opportunities for behavior versus physiological adjustment.
Yeah. Yeah.
And the first response is that it's hard to use behavior.
Yeah.
First of all, animals are going down autumn.
Yeah.
It's going to get cold months and months later. So
Yeah.
how do they predict
Yeah.
how cold, so on. And also, because Q10 effects, change in temperature has very little effect on metabolism per se. If you drop 5 degrees, that's not going to save you much energy.
Yeah.
So, just wondering, like in really cold environments for long periods of time, the best option is adjustment,
Yeah.
not behavior.
Totally.
For me as a behavioral ecologist, it's very hard to.
Well, that's not to say. So there's been some really interesting work recently coming out of Michael Dillon's lab on burying depth, and that there's been some evidence that there are latitudinal declines so for species that overwinter in the soil, they actually shift what depth they bury themselves at. So there is some behavioral choice in where they overwinter. The same is true for some species of budworm. Some of them, my guys actually overwinter on like the branches themselves and get nailed with the full extent of cold. But there are some species that overwinter on the bole of the tree, which is a little more thermally buffered. So that choice of overwintering site is really important. On the sort of Q10 effects, one of the interesting things to remember, and this is where that point you made in your talk about time is really important. Even if it's just a few degrees Celsius, when you integrate that over 6 months, that can actually really add up. But this is also where we think this fall warming is super important, right? Because at that point, they are so high on, their metabolic rate is so high already that, a couple of degrees of warming in the fall can actually really drive things as well. Yeah. So, yeah.
I have a question, kind of a follow-up
Mhm.
on that, which is just broadly how you think about this trade-off between behavioral and physiological response, because it seems like once you commit to current, you lose the ability to respond.
Yes.
Behaviorally, and a specific question related to that is, when you're looking at this kind of mortality due to fall warming, do you ever see cases where like a late fall heat wave is actually killing these insects because they're hitting like the upper thermal range?
We think so, but it's all very anecdotal. So the best data we've got on it is that paper that I mentioned. So this one suggesting that fall warming is particularly bad for butterflies. But yeah, I think that's a complete unknown for sure. So yeah. Yeah. Oh, sure.
I was curious how long glycerol is actually maintained in the body as a organism. Does it affect more fluctuations leading to overwintering? Is it just every time it gets sufficiently cold, they take a little bit of glycerol and then it hangs out for a long time, or are they doing something that's different?
Yeah. So In my study, to give you a little more background, I actually manipulated the number of exposures, the intensity of exposures, the time between exposures. So it was like, again, one of these like horrible experiments that you can only do with insects, or I guess with phytoplankton. Yeah. And what I found is that generally glycerol stuck around for a while. You would see this additional glycerol accumulated with 3 or more exposures, and that it seemed to be kind of entrained. However, if it goes long enough, yeah, there was a time in which they started like they can actually stick it into gluconeogenesis and kind of accumulate energy reserves from it. So yeah, and then of course in the spring, as things warm up, metabolic rates go up, and then they're just kind of using everything they can to get out and active. So yeah. Okay.
All right, thank you.
No worries.